From Microbe TV, this is TWiM, This Week in Microbiology, episode 228, recorded on October 15th, 2020. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schechter.
Well, hello there. Nice to be here.
And we are, for the first time, doing Zoom. For those of you who are just listening, you might want to check out the Zoom, because now we can see each other, and I get to see Elio. Nice to see you. I haven't seen you in 18 months. That's the last time we were out there.
I haven't changed a bit.
You haven't? Yeah. Also joining us from Ann Arbor, Michigan, Michelle Swanson.
Hello. Great to be with you all.
Are you out in the woods today there with a lake behind you?
I am. Yeah,
Nice.
The lake is not that close to me. That was a recent hike I did, though.
It's beautiful. Very nice.
Looking north up Lake Michigan.
Great. And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. Good to see y'all.
Actually, you can see us, but not the listeners, obviously. So,
Yeah.
Michael, you have a nice picture of Charleston behind you, right?
That is the Ashley River, and
Nice.
together with the Cooper River, it comes to form the Atlantic Ocean, according to what the locals tell me.
All right.
I don't know whether or not it's true or not. The Atlantic Ocean seems to have a lot of water, and we get a lot of rain, but not that much.
We have a special episode today. We have two guests. Our first is from the University of Alabama in Birmingham, Ninecia Scott. Welcome to TWiM.
Thank you guys for having me.
Pleasure. And from the University of Michigan, Chelsey Spriggs. Welcome.
Thank you.
I guess Chelsey and Michelle, you know each other somewhat, right?
We do.
Hmm?
Yep.
Yeah.
Hi, Michael. I can see you from Birmingham. We're kind of
That's true. It's only a 6-hour drive.
Right, if I go like this, I can see you a little bit.
Yes. We're representing the South.
Yes.
So Ninecia and Chelsey are both postdocs at their respective institutions. And you are you among the co-founders of Black in Microbiology? Is that correct?
Yeah, we're team leads. I'm the sponsorship lead, and Ninecia is one of the programming leads.
Okay. So yesterday on TWiV, we had two of your colleagues to talk about it as well. We're going to go through TWiV and TWiM and TWIP and have representatives from Black in Microbiology. So
Would that have been Ari Kozak from the University of Michigan?
Ari
Yep.
Kozak and Kishana.
Taylor.
Yeah, I can't remember her last name. What is it again?
Taylor.
Taylor, Kishana Taylor. I had to have help pronouncing her name as well, too. So great to have you. And let's talk a little bit about Black in Microbiology. And how did you get involved with it? Let's start with Chelsey.
Yeah. So Black in Microbiology is a program that we started this summer, really maybe end of July, beginning of August, to put together in response to some of the Black Lives Matters discussions happening this summer, and especially with academia and just the acknowledgement that there's not a lot of Black scientists in departments and institutions around the country and in the world. So there have been several of these Black in X movements, like Black in Neuro, there's Black in Cancer right now. And I know Kishana, I met her last year at a conference, and we were just like, oh my gosh, another Black virologist. Never met you before. So she actually texted me in July, and she asked, hey, I want to put together like a Black in Microbiology. Would you be interested? And I said, absolutely. So that's how I got involved with Black in Micro.
Yeah, yesterday Kishana said she didn't know any Black virologists. And she, after starting this, she says, it's amazing how many of them are out there. And now we've connected. And I think that's one of the great parts. How about you, Ninecia?
I guess I'm a little different from all the virologists that surround us in Black and Micro. I'm a bacteriologist. And so I met Kishana last year at Abercams, which is a ASM sponsored conference for undergrads. And there we kind of were talking. That's where you meet scientists at every level pretty much. And it was great to see that type of support. And so I followed her on Twitter a little time before that. And so we saw Black in Neuro, we saw Black birders, we saw all these other Black in whatever specific area or field there were. And I was like, Kishana, please say we're going to do a Black and Micro week. And lo and behold, she did. She put the call out there. And I was like, yes, I will definitely help to do this. And she was like, sure, Ninecia, we will love to have you. And so I'm grateful for all the people that I've met through Black and Micro as well.
Yeah, we heard yesterday that you had a week of talks. You had a roll call where people announced themselves on Twitter, and that is a resource that's going to be available it's already on Twitter. You can search for that. What's the hashtag that you search for? Black and Micro.
You can do Black and Micro. You can do BIM roll call.
That's great. And then you can
you'll get
all these people, hundreds of people who said, "Here I am. I'm a microbiologist. Let's interact."
It was really an exciting week because different fresh faces would pop up and they'd have these amazing expertise. And it was really just that you could feel the enthusiasm through the Twitter feeds.
Yeah, I thought it was really amazing too because the roll calls had undergrads, grad students, postdocs, faculty. And so you just get to meet people from all different levels of this journey. And, you can find mentors for yourself based off of where you are and where you want to be.
And the other thing that I think that was very powerful during the week was that we weren't just getting academics. We were getting people from industry and government and, policy, and just showing the breadth of where microbiologists actually can go and be beneficial within society, right? So we traditionally think that's in academia most of the time, but that's not true. We're diversifying everywhere and we're beneficial everywhere.
Yeah. You had a session devoted to career opportunities or career jobs for microbiologists, didn't you?
Yes, we did. So there were a couple of, as the programming leads, Linnell and I, we met for about a week and we sat and discussed. We said, okay, we have people's attention right now. What do we want to focus on? And so we knew the focus was, of course, highlighting Black microbiologists in these different fields, but we also wanted to break down a couple of barriers that are misconceptions that people had in terms of microbiologists. And so we highlighted different careers that microbiologists were actually having. And then we also incorporated STEM education, which was something that I don't think any other week had done in terms of trying to address the pipeline issue of how do we get more Black faces in micro.
So this that week was a great way to see to showcase the science in Black microbiology. But obviously, you don't want this to be a 1 week phenomenon, right?
No.
Are you being helped by the ASM?
Yes, actually, they're one of our gold sponsors, and ASM actually went as far as to try to establish a long-term partnership with us. So we'll be working with them in the future as well, which is really exciting.
It's terrific.
Very
It's
good.
a win-win situation.
Mm-hmm.
So
ASM has a long history of supporting Abercams.
Mm-hmm.
And when I was involved in the early days with Cliff Houston setting it up, Cliff is a microbiologist from Texas. And Cliff was chair of the education committee back in the mid 90s. And Cliff ultimately became the president of the society. And he was very much high on it. Cliff happens to also be Black, and he was always making opportunities to encourage and find folks and help them aggregate so that we wouldn't miss the opportunity to interact with our colleagues.
Yeah, we also have sponsorship from ASV, and hopefully in the future, maybe at one of those conferences, we could have Black and Micro programming, or just a space for us to come together, because a lot of us haven't met in person ever. Like, I've met Kashana, I've never met Ninecia, although I feel like I know you. Most of the organizing committee has never met each other. We've done it all over Zoom and through Slack, so it'd be really great to all have a space to come together, and I think one of those conferences would be perfect.
Well, someday I hope we do have in-person conferences again.
Yeah.
I'm not sure when, but that should happen. So, for you, what do you do going forward so that it's not a 1 week event? What has to be done to keep the momentum, so to speak?
I think Oh, go ahead, Chelsey.
Oh, I was just going to say, I think that, just making sure that we stay active on Twitter and engaging people, putting together our I guess, directory, and using our website as a resource for other people. Future programming, which I'm not sure what that would look like, but maybe we could have like a miniature conference, because we got to talk a little bit about our science in the roll calls, but it'd be nice to have like kind of a space for us to present our work and get feedback from each other too.
Mm-hmm.
I think those things would be nice.
Yeah.
What were you going to say, Ninecia?
I think so yeah, from the programming perspective, it seems like this was a rollout for us just to make sure people knew we were here, and knew our faces, knew our locations, knew our science a little bit, which was this is why the impact of the roll call was so important. So you're saying your name, you're saying what institution that you're at, and you're saying what you work on. But just going a little deeper to show that, okay, we do have science that we work on daily, and, foster this community that we have in addition to collaborations, right? So when we present our science, we get new perspectives as well as potential collaborators. And that's something that I think a lot of us are looking to develop.
It certainly takes work going forward, right? It's not going to happen automatically, but here on TWiV and TWiM and TWiP, we will go to that list from Twitter and have guests periodically on our shows. So I made that commitment a while ago, and I think we have quite a reach. So that would be one way we can help out, and also get good science, right?
And if I could interject, it's terrific that ASM, ASV, a number of journals, academic departments have been sponsors. But I saw from your website that you're also happy to take donations from individuals. So I would just make that plea to the people that are listening, our loyal listeners across the world, to log on to the Black and Micro. We'll put the link in the show notes, and you too can make a donation to support the work, because these postdocs and students that have been driving this, they're at a really critical time of their careers as well, and their personal lives. I know many postdocs maybe are setting up a household with a partner for the first time, maybe welcoming their first or second child, and doing their research and doing this really important outreach. So first of all, good for you, but also the rest of us need to step up and contribute.
Absolutely.
Thank you.
Don't be shy. People will donate for sure. I told the folks yesterday on TWiV, you should set up a 501(c) nonprofit and make a real organization, so you can hire people to do the bookkeeping and the organizational stuff that you don't need to do yourself. It can be done for sure. Ninecia and Chelsey, is there anything more about Black and Micro you want to mention before we chat a little bit about you two? Did we cover all you wanted to talk about?
I will say, right now we're in a phase where just because the Black and Micro Week is over, it doesn't mean that, we're not still active. So please come and join our community, right? And you don't have to be Black to join, right? You can be of any creed or color to join and support our community. Mm-hmm. So we are welcome to any and all, and we would be grateful to meet you.
How much work are you doing with other groups like Latinos, Native Americans, and so forth? Is there a connection with those groups?
So far, we haven't established any official connections, but I mean, we would love to work with all types of underrepresented groups. And if they're interested in doing a program that's similar so that they can find each other, I would be happy to help provide resources and advice. But yeah, all minority groups
Right.
are welcome, and we would love to work with them as well.
One thing I'd like to interject is to reach out to the ASM branches through a contact point of Ann Dempsey. If you write that you have formed this group and ask Ann to send it to the 35 ASM branches to announce your group, it's another way to recruit undergraduates who are, first dipping their toe into science, to know that they don't have to just go into medicine or dentistry or pharmacy school. They can be real card carrying scientists, and they're not the unicorn. There are many of them out there, and Ann's a good resource at ASM to contact if you have a generic notice that you want to send out to the branches. That's a good way to reach out and touch folks. We're having a branch meeting tomorrow in South Carolina, so I'll be certain to give your hashtag out to all the South Carolina students and postdocs who are attending that event.
Yeah, that's actually a really Oh, yeah, that's a great idea because most of our reach so far has been on Twitter. And so I fear that if people don't have Twitter, I just got a Twitter last year or something,
Mm.
they might have missed this whole thing. And so we want to be able to reach as many people as possible. So things like that where someone can get an email or people in the department can say, hey, connect with these folks through their website,
Right.
and hopefully when we get our directory up, we can have our contact information and people can just reach out as they see fit.
Mhm.
And if I could just say a shout out to Catherine Yu, a writer at the New York Times. She did a feature on the Blacks in Micro program, which was
Wow.
fantastic. So Twitter or the New York Times, they would have heard about it.
Yeah, it was a
Let me
great.
ask you another question. Is there any attempt to gather the historical facts? There are quite a few Black microbiologists in history, some of them which are very distinguished. I count among them a friend, Harold Amos, who was no less than the chair of microbiology at Harvard Medical School. Not bad. He was a fantastic guy, and we wrote him up recently in our blog. Anyhow, the fact that there is a history behind it, and that there are very distinguished microbiologists who happen to be Black, I think is worth noting.
Yeah, and that's very, very much true. So our last day of Black and Micro Week, we had the hashtag micro racism and history of micro. And so that was geared towards understanding our past and where Black microbiologists actually intersected with science and how we propelled science forward in microbiology. So hopefully in the future we can drop more gems like that, because I think a lot of people were surprised by the people that we had on our historical figures list.
Exactly.
And they were like, "Oh, I didn't know this," or "Oh, it goes as far back." Black influence in science, and specifically in microbiology, goes back this far. Yes, we're a part of the community. We're active in trying to expand and move the field forward, and so we'll continue to do that.
Good point.
Really.
So let's hear just a bit about yourselves, Ninecia. Where are you from originally?
I'm from Raleigh, North Carolina, so if I were at home, I really could see Michael from my backyard. I went to undergrad at North Carolina Central University, which is a HBCU, and I got my degrees in biology and pharmaceutical science. And that's actually where I met my first Black microbiologist. And so from there, I actually worked under Dr. Daniel Williams for a majority of my undergrad on Neisseria gonorrhea, looking at antibiotic resistance. And from there, he kind of encouraged me to do summer programs. So I went to Wash U, I went to UCSF, I went to Duke, doing fungal and bacterial work. And then I decided I had so much fun at WashU in St. Louis that I went back for grad school.
All right.
And so I got my degree in molecular microbiology and microbial pathogenesis, where I studied tuberculosis, specifically neutrophils and how it shaped acute and chronic infection. And then I fell in love with neutrophils and lung biology and bacteria. So I decided to pivot a little bit to Streptococcus pneumoniae, which is what I do here at UAB. And we're looking at to try to understand the dissemination of Streptococcus pneumoniae throughout the body.
Can you remember how far back in your life you decided you wanted to do science?
My goodness. I think it was very much fostered by my mom and my dad. So they weren't, and so I probably had to be in elementary school. They were always the type of parents that would say, "Look it up, Ninecia. Why is the sky blue?" I don't know, look it up. And so I would actually have to, and I mean, I know if some students are watching this, I had encyclopedias. So I had to go to the encyclopedia and like look up sky and look up blue, like what is blue? And so, cause we didn't really have internet back then. I'm aging myself, but we really didn't. And so, my parents really fostered that. And then throughout my life in middle school and high school, I will say that North Carolina has a very rich history in education. So they had NSF funding programs through NC State that I went to, learning about photonics and Infotap, which was another Black physician and scientist, just trying to encourage underrepresented minorities to go into science. And then it peaked when I went to UNC Charlotte. I was given like a scholarship in high school to go there, where I learned about antibiotic resistance and MRSA. And I was like, I'm set. I'm going to do bacterial work. I'm going to figure out an antibiotic that's going to cure everything.
Hey.
But I've come a long way from that goal. But
go for it. Go for it.
You'll never be bored.
Literally. But yeah, it's been a long time, probably since childhood.
Mm-hmm.
Oh, wow.
And what do you think is next for you after your postdoc?
After my postdoc, well, I'm taking a step at a time right now. Trying to get the next paper out. I just recently had my JCI paper published from grad school.
Nice.
All right.
So I'm working on the paper here from UAB. And after that, a faculty position at an R1 institution.
Do you like teaching?
So actually, that's an interesting thing that you say. So I actually came to Birmingham as a Aracta Fellow. So as an Aracta Fellow, you teach at a HBCU. So right now I'm teaching microbiology and slightly vaccinology at Oakwood University, which is a HBCU here in Alabama. And you teach while you do your research. So I was on the Aracta Fellowship my first year, and then I transitioned to an immunology T32 this year. And but I'm still teaching on the side.
Great.
So you're a juggler.
I try my best.
Juggler.
I try my best.
Yes.
Chelsey, where are you from?
My family is originally from DC, but I was raised in Detroit, so not so far from here. They're all in Detroit right now, so
Great.
I love it being so close to them.
Where'd you go to college?
I went to undergrad at Michigan State in East Lansing.
They've got really strong undergraduate microbiology.
Yeah, so I was a microbiology major at Michigan State, and I guess that's kind of how I stumbled into virology. Is I was looking for a lab to work in for undergraduate research requirement, and I emailed Dr. Michelle Fluke, who was there at the time, and she studied polyomavirus as a model for breast cancer progression, and I was like really interested in cancer, and I thought that was just such a cool way to use viruses as a tool to learn more about this really important disease. And actually, I was thinking about something that Michael said about Black students not knowing that they could be scientists and thinking about like medicine or veterinary medicine. And that was totally me. I thought that, okay, I like science and I'm smart. I have good enough grades. I guess I'll go to med school. Um, and even though I was doing this undergraduate research, it still seemed like, okay, yeah, but I still supposed to go to med school. That's what I'm supposed to do. So I applied for an MD PhD program at Morehouse College in Atlanta, and I went there. And I just kind of thought, this is okay, but I can't wait till the research part starts in 2 years. Like, wow, can we get through this med school part? And then something clicked, and I was just like, I should just go to get a PhD. But yeah, I hadn't even learned about grad school until maybe my junior year in college, but I was like full swing into like the MCAT, and like that was my path. So I called Michelle and I'm like, I gotta leave. I want to go to grad school. I need help. So she helped me with everything. She told me, oh, you don't have to do a master's. You can apply straight for PhD, which I didn't know was a thing. She helped me with my applications and she just really set me on this path. So I ended up going to Northwestern to do my PhD, and I worked with Dr. Lou Lamons there, studying the DNA damage response in human papillomavirus replication and transformation, and that was awesome. I had like a really great experience at Northwestern and in the microbiology department there. And then when I transitioned to postdoc, I wanted to move back to Michigan if possible. And I was looking for labs and was like, hey, do you know this guy named Billy Sy? And I hadn't seen him on the website, but they were in study section together. So it's this whole like connection. I think Lou also knew Michelle from like conferences. So
Yeah.
um, it's
And
kind of
Billy
cool
was
how
really
everyone
trained
just
as a cell biologist, and he's now moving to the
exactly.
interface. So it perfectly complements your training, undergraduate and
Yeah,
graduate training.
And you need
exactly. And
not
he's
be ashamed of leaving medicine for the great world of science. What you need to tell all your friends is that doing an experiment and seeing the results in the lab, it's more addictive than surfing. And if you know anything about surfing, people will tell you it's more addictive than the most addictive illicit drug.
Oh, wow.
And when science works, it's addicting.
That's true. Yeah, because I feel like science does not work a lot. Like the amount of experiments that fail versus the ones that
Oh
go
yeah.
right, it makes
And
me
that's
addictive
why
for
we
us to
call
like
it
sit here.
research and not search.
Again, that's true. I'm
Chelsea,
like,
what?
shit, I always tell my mentees, we do research because we have to do it again and
Yeah,
again.
that's right. So, Chelsea, what are you doing in the lab at Michigan?
Right now, I'm studying how polyomaviruses get into the nucleus during viral entry. So I've been on this like non-enveloped virus, DNA virus path, and so it's kind of cool that I'm back studying polyomaviruses when that's where I started. Um, so yeah, I published a paper earlier this year showing how the virus gets disassembled in the cytosol using some dynein motor machinery. And so now I'm like looking at the next step of how it actually gets into the nucleus. So starting a new project, which is kind of cool and kind of scary all over again.
Great.
And your funding is coming from
Oh yeah. I had a T32 from the micro department. I was on that for a year. The molecular mechanisms of
mechanisms.
microbial pathogenesis grant.
Yep.
So that was really great because I'm in the Department of Cell and Developmental Biology at Michigan. And I love this department. It's like so awesome, but there's not really much microbiology there outside of our lab. So getting to integrate a little bit with the micro department has been kind of fun also. And right now I'm on an F32 grant and I'm hoping I get my K99, which I applied for in June. So
Yeah.
Very good.
Yeah.
And
Chelsea is paving the way.
what is next for you, Chelsea?
so right now I'm focusing on my science, but also just trying to get the training that I need to move forward for finding a faculty position as well. Just applying for all the grants. Oh, I also am on the Burroughs Wellcome Fund postdoctoral enrichment program fellowship, and that one's awesome. Just getting to connect with different scientists, and they are so good about promoting diversity in science, and I'm really excited to be a part of that one. But yeah, just working on science, trying to get this next paper, and then hopefully moving on to a faculty position.
So,
Well, could I also say that we know from polling employers of our PhD trainees at Michigan that they're also looking for people that have exhibited leadership and know how to lead a team. And so the work that you've done through Black in Microbiology is really going to be a beautiful jewel for your CVs or your resumes. So the fact that you're accomplishing a lot at the bench and being successful there, and then also have these teamwork skills, leadership skills, and development skills are really going to be give you a leg up. So good for you.
Thank you.
can
Thank
you
you.
can you remember how far back you were interested in science?
Oh wow. Let's think. I think probably middle school. My mom, like Ninecia's parents, she was very into like the science fair. I don't know why, because she's not a scientist. She does business. She worked for Ford, but she would be all gung ho, like, "Oh, I read this thing online. We gotta figure out an experiment where we can kind of test these things." Like I had this science fair project where we looked at electricity in plants and how it affects plant growth, because she read that like these giant trees are forming in the forest and it's like electricity strikes the ground however many times. And so she's like, I know what your science fair project's going to be. And so we had this whole elaborate setup. And then I think I'm really motivated by positive reinforcement. And so I won the science fair and I was like, ooh,
Nice.
I won. I should keep doing this. So I think ever since then, I've just been like really into science.
Wonderful. Well, I wish both of you the best of luck in your careers, and hope that you get everything you want. And we're very impressed with Black in Microbiology, what you've done, and hopefully it will keep going forward and inspire many, many young people to enter the field, because that's what you need, right? So our guest today from University of Alabama, Ninecia Scott. Thanks, Ninecia, for joining us today.
Thank you for having me.
Pleasure. Look forward to seeing your paper here on TWiM one day.
Oh, hopefully.
And from the University of Michigan, Chelsey Spriggs. Thanks for joining us, Chelsey.
Thank you.
And we'll look forward to your paper on TWiV, I guess, right?
Yeah, viruses.
We do have someone on TWiV who likes DNA viruses, so
Nice.
that would be...
Would that be Kathy?
Yeah.
Or would that be Rich?
Yeah, two people. I'm thinking of Kathy because they're all at Michigan, but we like all viruses, actually. Anyway, thanks to both of you for joining us. We really had a good time. And good
Thank
luck.
you so much for everything you've done for our community.
Thank you.
Bye-bye.
Thank you.
Bye-bye.
Bye.
All right. What great.
It was neat.
Great.
That was delightful.
Just delightful.
That was lovely.
It's going to be a tough act to follow, Elio.
Oh my God.
And
But
Elio,
it's easy because I'm in outer space.
You're in outer space, the
I'm
final
in outer
frontier.
space. So the paper that I'm going to discuss is by no less than 17 Japanese scientists. The first named one is Yuko Kawaguchi, and the last two are called Shinichi Yokobori and Akihito Yamagishi. And they are from all over Japan. In fact, they're from places I never heard of. The universities at Hachioji, Oragun, Ikabari, Ikaraki, Sagamihara, Nagaoka, Chiba, and Fukuoka. So you're taking a trip through the geography of Japan. So what's this about? I can tell you the punchline very easily. These are people who are studying the survival of bacteria in outer space by attaching them to the outer surface of the International Space Station, where they can retrieve them at different times and determine their viability. The punchline is the following. It's been known that bacteria can survive in space if shielded from ultraviolet light. That is, if they're put in a capsule which shields them, which avoids the ultraviolet light, they can survive for quite a few years. That's a known fact. It's been studied by several labs, and the organisms are mainly Bacillus subtilis spores, because when you think about survival under strange conditions, spores come right up because they're, of course, the toughest form of life. Now, that's all good and dandy, and you can derive from that the fact that bacteria can survive in space for long times. However, the question is, does it take a shielding material? They talk about small fragments of rocks, or oddly, I don't know where this comes from, mixtures of sugar or clay, sufficiently thick to protect them from irradiation. Now, why is this interesting? It's interesting because for a long time, people who worry about simple things like the origin of life have been wondering whether the origin of life isn't something that happened in space and was transported, forms were transported to this Earth by essentially meteors or micrometeorites. This is called the panspermia theory, hypothesis, and it's popular in some quarters. Some people believe that a way out of understanding how life started on Earth is to say it never did start on Earth. It started someplace else.
And
I'm not so sure that this isn't just shoving the question.
In fact, Michael Crichton had a very famous novel that effectively addressed this hypothesis of panspermia that many of our listeners probably read as teenagers, called The Andromeda Strain. And it's a really fantastical book that really addresses this whole notion of space and interplanetary transport.
All right, so this is what they did. The Japanese own part of the International Space Station. They have a chunk of it, which they call, they have a part of it, they can apparently do whatever they want with it. But of course, it takes a collaboration of everybody in the space station, which of course they used. So their team goes under the name of Tanpopo, which means daffodil.
Dandelion.
Dandelion.
I'm sorry, dandelion in Japanese. So why dandelions? Because dandelions send out the seeds far afield. So they call it dandelion, and dandelion, Tampopo, is a way in which they can do the study. So the study they do is with an organism called Deinococcus radiodurans. Radiococcus radiodurans is almost as good as spores in the sense that it's extraordinarily resistant to radiation. Radiodurans means that. And Deinococcus means mighty coccus. It's an organism that's been found in the affluent from atomic power stations, because there's a lot of radioactivity being spit out, and these organisms just laugh at it. They are totally resistant to it. So you take Deinococcus and you put it in little containers, and the containers have to be very special and very specially done. And there's a lot of technology here. So if you like to read about how to do things, this paper is just absolutely full of information. If you want to reproduce Tampopo, you can try. You have to have a chunk.
But you need a rocket. You need a rocket, Elio.
SpaceX.
Rockets are a dime a dozen now.
Okay.
You need cash.
Anyhow, if Deinococcus, so what they do is, instead of shielding the Deinococcus cells by using a metal or some kind of a radiation shielding device. They say, let us see if they can shield themselves. That is, if you have a collection of Deinococcus, a layer of Deinococcus, which is, let's say, a tenth of a millimeter, half a millimeter, 1 millimeter thick, what will happen? But when they found out, it's very simple and kind of predictable. The outer layers are dead, are going to die, because they are the ones that are not shielded by anything. However, the dead cells stay there, and they shield the inside cells from radiation. So boy, does that work. It works so beautifully that they calculate that if you have a 1 millimeter thick layer, this will go on. They will survive, by their measurement, 3 years in space. They can be retrieved then, and you find that there is damage, but Deinococcus is very good at repairing the damage. And how do you know that? You know that because you can do the same experiment with bugs which are mutant in some radiation systems. And they found the radiation systems that are involved are called excision repair, where you take a chunk of DNA which has been damaged, take it out, and put a good one in. So that's the conclusion, that in fact, if you want to have bugs which may survive, who knows? Certainly 2 to 8 years is the experimental thing here, but it could be easily 50 years, 100 years, something like that. You can then count on the bacteria providing their own shielding. So that's the conclusion. That's about the message. As I say, there's a tremendous amount of technical detail, how you do the experiment, how do you set up these little containers and so forth. But that's the conclusion. These bacteria, and by the way, it's not just radiodurans, but they also found a couple of other Deinococcus strains, which they sampled from the stratosphere. So if you go up in space or near inner space, and you find bacteria there, and some of them are Deinococcus of different kinds. Okay, that's the story.
I also appreciated that they kind of created an image for us that some microbes, of course, are growing really in close association with rocks or sediment particles, and so that would be a way for them to form these consortiums and provide more of a barrier to the UV damage.
That's been known. I mean, let's say, and they gave this a name, lithopanspermia.
Panspermia, yes.
Yeah.
And then since there's no litho in their experiment, they call it what do they call it again? Massa panspermia, massa for mass. That is, just providing enough mass is a way of shielding from UV. But everything is UV here, because UV is the most obvious thing. But in reality, there's lots of things going on in space, which even if you are shielded from UV, are damaging, like vacuum,
gamma rays.
That's right. Ionizing radiation of different sorts, and so you can really say that there is a lot of stuff, that desiccation itself, drying out. So anyhow, Tampopo is a good way to study such things.
So Elio, does this prove that life came from another planet?
It does not. It says life could come from another planet. It might, perhaps. It does no such thing as prove it. Thank you for asking that. In fact, the world is divided in believers and non-believers. I mean, there are people who are very panspermia oriented and people who say just another hypothesis. It's at best a hypothesis.
As they point out, they have to have been ejected from a planet. They had to be transferred somehow. Then they have to land. And these are not trivial things, right?
And landing generates autoclave-like temperatures.
That's right.
And, a number of years ago, there was a paper in Science effectively advancing this hypothesis that said that one of the ejected fractions from Mars that landed on Earth actually had fossilized bacteria in. And I never followed that story all that closely to find out whatever became of that. But as it was not in here, I think it probably died on the vine.
I imagine it did, yes.
Well,
I think this is a nice piece of work. When it comes to the origin of life, anything goes, right?
Well, I still remember Kate Rubins, the astronaut, talking talking on TWiM at an ASM meeting, and she was describing how many great lengths the astronaut teams go through to conducting these sorts of experiments. And one of the things that was interesting in the methods is that Mark Kelly, the human who has spent the most time in space, was actually the individual who went out and placed the canister on the side of the International Space Station.
Scott Kelly.
Scott Kelly.
For today's paper, Michael.
Yeah.
Okay, it wasn't Mark?
Well, today's paper is Scott.
Okay. All right, then I misread or misremembered.
Is there a Mark Kelly also?
Quite a coincidence.
They're twins.
Yeah.
They're twins.
Oh,
There's a Scott. Both of them are astronauts, and both of them went out to the space station.
But not all of them are running for office.
Not all of them are running for office.
So
that's
Kate,
right. It was Scott Kelly. It wasn't Mark Kelly.
Kate Rubins is back up. She just went back up.
She did?
She's back on the ISS for 6 months, yeah.
Oh, really?
Wow.
Doing experiments,
Good
yeah.
for her.
I wanted to just say that there is a very nice line of evidence that life, in fact, began here on Earth. And many people work on it, and there's a good sequence of events that are plausible, some of which are supported experimentally. Whereas, I mean, this assumes Deinococcus existed somewhere else, and maybe it didn't, and maybe something. I think it's curious, but I think a lot of things have to happen, and I'm happy to entertain the notion that life, in fact, began here, because I think it beginning here is more fascinating than it coming from somewhere else, right? That's
Well, the thing is, if it comes from somewhere else, it happened by some mechanism.
Sure.
See, it's not like it happened out of nothing.
Right.
In any event, you have to postulate a series of events that led to the formation of something living. The problem is just as hard on some planet out in space than it is here.
Yeah.
So you're not gaining much.
Sure.
With panspermia.
Sure.
It doesn't help. It doesn't facilitate the process.
Another implication is for when we send ships out into space, that we need to be careful about not introducing inadvertently microbes that could survive.
Yeah.
Right? I think NASA goes to great lengths to avoid contaminating.
Sure.
They do indeed. They scrub things, and of course, that's one of the positive controls they have to be able to be sure about when we begin to go looking on Mars for remnants of life.
But didn't we introduce bacteria to the moon, right?
Oh yeah.
And how?
We did.
We left behind stool. We left behind stool.
Okay. Yeah, we did.
And not the three-legged type.
No, not a three-legged stool.
Elio, Deinococcus is highly radiation resistant. What's selected for this? Where does it inhabit that this property was selected for? Do you know?
I don't know, but I think maybe Michael knows, but I mean, it's found in places with high radiation.
Like high altitudes?
So you assume that they're selected for that.
Okay.
For survival under high radiation conditions.
They have a very high replication rate, so they have multiple replication forks on their chromosome.
Right.
So they evolved a very sophisticated repair mechanism to deal with these multiple replication forks. And they're in continuous compare and contrast of looking at the daughter segments that are being produced, asking fundamentally, how is it different than the other one? So the selection pressure was probably for rapid growth as opposed to being UV resistant or something. And I think the radio resistance is likely happenstance. It was probably optimized for competition to grow quickly.
Got it. And do you find these all over the planet in soils and so forth?
It's very common.
Yeah.
It's very common.
Okay.
And this guy's finding another couple of species is kind of interesting.
And my last question, do these ever cause human infections?
No.
No, we're not that radiation prone.
We're not ready. I get sunburned bad, so it's not.
Got it. All right. I have one email which I think will be interesting. This is from Scott, who writes, "Dear TWiM podcasters and listeners, after your fungi-centered TWiM 227, I wanted to share this virtual seminar series from world-renowned medical mycologists giving talks on fungal infections and immunity to these diseases. The Center for Medical Mycology at the University of Exeter in the UK, in lieu of in-person meetings, has assembled a virtual seminar series of some of the best research of medically relevant fungi." And Scott provides a link. The series is a full year of monthly seminars featuring 2 distinguished researchers. Past month seminars have been posted on YouTube and can be reached on the site. You can also sign up to view the live upcoming seminars. Scott is a postdoc at UW-Madison, University of Wisconsin-Madison. Thank you, Scott. That's a great resource.
It sounds like appointment listening for Elio.
That's right.
Yes, well, I should.
You should.
I'll look into it.
Okay. All right, that's TWiM 228, our first Zoom TWiM. You can find the show notes at microbe.tv/TWiM. If you want to send us questions or comments, you send them to twim@microbe.tv. And if you like what we do here on TWiM and on the other podcasts, consider supporting us financially. Go to microbe.tv/contribute. Michelle Swanson's at the University of Michigan in Ann Arbor. Thanks, Michelle.
Thank you. Great to see you all.
It's good to see you. And it's easier to talk without stepping on each other when we can watch each other talking and you wait for them to finish, right? It's really cool.
I think it's worked very well.
Elio Schechter is at Small Things Considered. Thanks, Elio.
Take care. Nice being with you. Nice seeing you.
Michael Schmitz at the Medical University of South Carolina. Thanks, Michael.
Thanks, everyone.
And I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.